Objective number 1, what environmental factors influence microbes and the functioning of their enzymes? Things like temperature, oxygen requirements, pH, osmotic pressure, and barometric pressure. And in this section, we're going to talk about temperature and oxygen requirements. There's a range of temperatures where microbes can grow, and that's called their cardinal temperatures. And some microbes have a very narrow cardinal range. Others have a very broad one. The minimum temperature is the lowest temperature that they can handle and still grow and carry on metabolism. The maximum temperature is the highest temperature that allows them to still have growth and metabolism. Optimum temperature is a small range. It's intermediate between minimum temperature and maximum temperature, and it promotes the fastest growth and metabolism, the fastest rate of growth and metabolism. We have different temperature adaptation groups. The psychrophiles have an optimum temperature below 15 degrees Celsius. They can grow at 0 degrees Celsius in freezers. And examples here would be Staphylococcus aureus and Listeria monocytogenes. Mesophiles, their optimum temperature range is 20 to 40 degrees Celsius, and this is most of our human pathogens. Thermophiles grow best at greater than 45 all the way up to 80 degrees Celsius, and this would be your archaea that are found in hydrothermal vents and geysers. This is a graph of the different types of adaptation groups for temperature. The psychrophile is the blue here, and you see it's at below 15 degrees Celsius, below it. The psychotrophs, these are the ones that grow slowly in the cold, and their optimum temperature is going to really be above 20 degrees Celsius. They'll grow up to about 35 degrees. The mesophile, which that's most of our pathogens in the green, is found here. Optimum temperature around 40 degrees Celsius. The thermophiles grow at better than 45 degrees all the way up to 80 degrees Celsius. And an extreme thermophile grows between 80 up to around 120 is its optimum. So they have adaptations that allow them to survive at different temperature ranges. Now, objective number 6 is here. These are the gas requirements. Oxygen gas is the one that has the greatest impact on the growth of the microbes. And as oxygen is used, it's changed into some toxic products. You've got the singlet oxygen, which is O2, and then you have superoxide ion, peroxide, and hydroxyl radicals. But most cells have developed enzymes to neutralize those toxins, and that would be your superoxide dismutase and the catalase. Now, there are different adaptations for their oxygen requirements. Aerobes have to have oxygen, and they can detoxify it. You've got obligate aerobes that are obligated to grow with oxygen. They cannot grow without it. Facultative anaerobes use oxygen, but they can also grow if they don't have oxygen. And a microaerophile doesn't grow at normal atmospheric concentrations of oxygen. It only needs a small amount of oxygen. So if the microbe cannot deal with toxic oxygen, it has to live in oxygen-free habitats. Anaerobes don't use oxygen. Obligate anaerobes, they can't detoxify oxygen, and they are obligated to live in an environment without oxygen. Aerotolerant anaerobes don't use the oxygen, but they can still survive and grow if it is there. And when you're growing anaerobic bacteria, you got to have special media and methods to incubate it and exclude the oxygen, because most microbes require some carbon dioxide in their metabolism. A capnophile grows best when you have higher carbon dioxide tensions than normally present in our atmosphere.